In order to implement longevity medicine approaches, it is necessary to have a good understanding of what aging is and its mechanisms. In our course, aging is considered as an exponential decline in homeostatic capabilities, leading to the increased risk of age-related diseases and death. At the organismal level, aging is impaired maintenance of homeostasis, system inflammation, intercellular signaling disorder, metabolic disorders, disturbance of circadian rhythms, an overall decrease in body functions and structures, loss of reproductive ability, and many other physiological features. An elevated level of teratogenesis in progeny. Aging leads to the development of age-related diseases and frailty. Conceptually, there are 4 different points of view on the basic mechanisms of aging. At the moment, the theory of programmed aging occupies a marginal position, suggesting that with age, special ontogenetic mechanisms are activated in order to eliminate an individual from the population and make room for future generations. Similar mechanisms, as supporters of this view suggest, can purposefully turn off the reparation and regeneration pathways. However, at present, humans do not know genes, hormones or organs that are evolutionarily predetermined to activate the aging program. The theory of biological imperfectness of regulators of homeostasis means that aging occurs as a result of the accumulation of metabolic errors, for which evolution has not provided effective mechanisms for their repair. This is the accumulation of crosslinks in long-lived proteins of the extracellular matrix. The concept of the quasi-program of aging, developed by Mikhail Blagosklany, provides that the continuation of the program of development of cells and tissues after reaching reproductive maturity leads to hypertrophy and hyperfunction, causing disruption of homeostasis. The most mainstream point of view is that metabolic errors are integrated over a long period, since some of them accidentally escape from the repair mechanisms, which ultimately leads to a systemic disturbance of homeostasis and the subsequent development of age-related diseases. As it is considered today, about 25% of life expectancy depends on heredity, for example, the risk of age-related diseases. Another example is mutations that contribute to the development of progeria and accelerated aging syndromes. The remaining 75% of aging depends on environmental factors, toxins, ionizing radiation, smoking, alcohol, and random factors. However, these are also factors associated with the work of certain genes, the so-called epigenetics. Hereditary and environmental factors together lead to the accumulation of metabolic errors. In the overwhelming majority, they are either prevented by certain intracellular systems, or undergo repair, restoration, and in the case of irreparable damage, removal, and replacement by newly synthesized macromolecules or new cells. Due to the insufficient efficiency of the systems of elimination of damage, homeostasis is disturbed, and the adaptive abilities of the body decrease, which is accompanied by an increase in the risks of age-related diseases from which the body dies. Changes during aging can be roughly divided into several levels. The first level is the accumulation of damage and errors. Here genomic instability takes place, which includes shortening of telomeres, transposition of mobile genetic elements, accumulation of mutations, and chromosomal breaks. In addition, epigenetic drift occurs, including chromatin remodeling, modification of histone proteins, expression of variant histones, changes in the level of DNA methylation, accumulation of epimutations, and changes in the expression pattern of non-coding RNAs. With aging, mitochondrial dysfunction is observed, which is accompanied by clonal expansion of mitochondrial DNA with deletions accumulated in mitochondrial DNA, accumulation of oxidized guanosines in it, a decrease in the expression of mitochondrial proteins, a change in the potential of the inner mitochondrial membrane, the opening of pores of temporary permeability, and release of pro-inflammatory factors from mitochondrial DNA. Finally, there is a loss of proteostasis, including the formation of crosslinks between molecules of long-lived proteins inside the cell and in the extracellular matrix, the accumulation of aggregates of oxidized proteins, an increase in matrix rigidity with the subsequent activation of the integrin mechanosensory response, the development of stress in the endoplasmic reticulum, inhibition of the proteasome and autophagy, and changes in the expression. In response to the accumulation of these damages and errors, a reaction of the cell and the body occurs, which leads to changes in the regulation of physiological processes, disruption of tissue regeneration, and systemic inflammation. As a result, systemic changes occur, including disruption of the functioning of the internal clock, chronic stress, increased permeability of physiological barriers, and metabolic disorders. Thus, a deterioration in the regulation of biological processes and a violation of homeostasis increases the risks of age-related diseases, decreases resistance to resistance, and increases the risk of senile fragility and death of the body. If we compare the functional capabilities of the human body at the age of 75 and in the middle age, in the range from 22 to 45 years, one can observe a significant decline in a large number of functions. In particular, the speed of nerve impulse conduction is reduced by 10%, the blood supply to the brain by 20%, and the blood supply to the liver by 30%. The rate of glomerular filtration of the kidneys decreases by the same amount, the vital capacity of the lungs drops by more than 40%, the rate of blood pH restoration is more than 80%. It should be noted here that aging is not only a process of reducing all kinds of body functions, some processes, on the contrary, are activated. In particular, morning levels of the hormone cortisol increase with age. In the blood plasma, there is an increase with age in the amount of pro-inflammatory proteins. Biomarkers of healthy longevity can be divided into three large groups: biological, functional, and digital. Biological reflects the length of a healthy period of life. This includes many different biomarkers that can be combined into separate blocks and analyzed using multivariate statistical methods. They can be used to measure and visualize the body's biological response to treatments and stressors. Functional markers often reflect the risks of senile fragility. Digital markers can be modified and captured using a variety of devices, including health gadgets and face photos. Here are examples of anthropometric and functional tests and age-related changes in parameters for assessing hand strength, body mass index, balance, fine motor skills of the hands, etc. Along with these parameters, there is a fairly extensive set of instrumental and biochemical studies that reflect the degradation of specific functional systems such as visual, auditory, cardiovascular, excretory, and neuroendocrine. Some of the biochemical markers of blood do not simply correlate with age but reflect the risks of overall mortality, such as the parameters of the lipid profile, glucose, lactate, some amino acids, albumin. Age-dependent markers of aging can be clustered into groups, including markers of inflammation, respiratory function of the blood, insulin resistance, dyslipidemia, endocrine regulation of reproduction, markers of the functional state of the liver and kidneys. Another cross-section of markers of age-related deviations at the level of the whole organism is the groups distinguished according to the anatomical principle, for example, markers of the state of bone, cartilage, and muscle tissue. An old dream of experimental and clinical gerontologists is the creation of an integral calculated indicator, the biological age, on the basis of clinical markers of aging. Deep aging clocks developed by Alex Javaronkoff in 2016. The aging of the arterial vessels is the most critical in terms of the development of age-related diseases and mortality. In the arteries, a thickening of the basement membrane is observed under the action of the end products of glycation and the smooth muscle layer of blood vessels, cross-linking or fragmentation of the extracellular matrix, activation and increased permeability of the vascular endothelium, deposition of cholesterol and calcium in the vascular wall. Arterial vessel age can be assessed by intermediate thickness estimation or pulse wave velocity measurement. Comparative analysis of the age dynamics of 86 aging biomarkers has shown that it is these parameters related to vascular health that have the greatest predictive power. The outstanding gerontologist and immunologist Claudio Franceschi identified the intermediate link between the molecular mechanisms of aging and pathologies as inflammaging. Inflammaging is a systemic, chronic inflammatory response not associated with infections. Its cause is the accumulation of defective mitochondria in cells, accumulation of senescent cells that produce inflammatory cytokines in tissues, and visceral obesity. Systemic inflammation increases the risk of virtually all age-related diseases, including cancer, neurodegenerative diseases, musculoskeletal conditions, lung disease, autoimmune diseases, metabolic disorders, and cardiovascular diseases. One of the most accurate molecular tests for biological age reflecting inflammaging, is the lactosylated N-glycans test for blood immunoglobulins. N-glycation is a post-translational mechanism regulating the function of immunoglobulins. The change in their condition is related to inflammation. The integrative measure of inflammatory processes, glycan age index, based on the analysis of blood combinations of the three egg glycans, including Fa2b, Fa2G2, and Fa2BG2, allows predicting the age of an individual with an accuracy of 9.7 years. External and internal causes of inflammation activate innate immunity and deplete the reserve capacities of adaptive immunity. As a result, chronic inflammation, depletion of hematopoietic stem cells in the bone marrow, a shift in the formation of the myeloid lineage, and involution of the thymus develop. Immunosenescence and inflammaging are formed, which increase the risks of infectious, oncological, cardiovascular, and autoimmune diseases, neuroinflammation, depression and neurodegeneration, and reduce the effectiveness of vaccination. One of the key reasons for the deficiency of naive lymphocytes with age is the aging of the stem niche of the red bone marrow, an increased rigidity of the extracellular matrix due to accumulated crosslinks and a decrease in the production of IL-7 by bone marrow stromal cells. An epigenetic shift and decreased expression of genes that regulate the lymphoid lineage occurs in the progenitors of lymphoid cells. As a result, more pro-inflammatory myeloid cells than lymphoid cells are formed with age. The formation of naive T and B cells decreases. Aging can be seen as a chronic stress response. Excessive amounts of stress hormones glucocorticoids promote fatty degeneration of the thymus, which leads to a deterioration of its immunoregulatory function and a decrease in the number of naive T cells, hyperactivation of dendritic cells, a decrease in the production of anti-inflammatory cytokines, and a decrease in the repertoire of T cell receptors. The profiling of age-related changes in a large panel of immunity markers made it possible to create a high-dimensional trajectory of immune aging, IMM-age. Possible strategies to enhance T cell immunity include the following: gene therapy for thymic regeneration, intervention in particular cytokine levels, influencing the precursors of immune cells, reducing fatty infiltration of the thymus, intermittent fasting. Another source of inflammaging is a decrease in the biodiversity of the core microbiome, an increase in the number of pathogenic bacteria, and a decrease in the number of beneficial ones. It should be noted that the microbiota of centenarians has its own characteristics and is more reminiscent of that of young subjects. The next